Okay, let's dive right into this explainer. We're going to uncover what is basically a gripping espionage thriller happening right now, literally inside your own biology. We're talking about a microscopic heist pulled off by some of the most notorious criminals out there, viruses and cancer. This invisible battle happening at the cellular level, it determines whether you catch a passing cold, develop a serious illness, or stay perfectly healthy. So yeah, the stakes are absolutely massive.
Think about this for a second. What if your body's energy factories were secretly working for the enemy? It sounds, exactly like a plot from some sci-fi movie, right? A hidden invader taking over the host's machinery. But this hostile takeover is not fiction. It's a biological reality happening inside our cells constantly. It's orchestrated by invaders that don't just want to survive, they actually want to weaponize your own biology against you. So cellular metabolism isn't just about digesting your breakfast or keeping you fueled up for a run. It is, in reality, a high-stakes biological battleground. When a pathogen, like a virus or a tumor, takes hold, they aren't just passively hanging out in your body. They aggressively take command of your cell's manufacturing plants to build their own armies. They literally turn your life-sustaining factories into weapons manufacturing plants for themselves. It's wild.
Chapter 1. The Microscopic Factory Heist. Infiltrating the supply chain.
Now, before an invader can rob a factory, we've got to understand the target. This factory produces everything a cell needs to survive. The whole shebang we call metabolism. Think of it as the ultimate hyper-efficient manufacturing plant. Inside, there are literal assembly lines of enzymes passing along nutrients like workers on a conveyor belt. They're tirelessly building the essential parts that keep you alive. It's honestly a masterpiece of biological engineering.
But then the heist happens. Normally, a healthy cell just goes about its peaceful daily routine, using resources like glucose to maintain balance and, keep the lights on. But when infected, those exact same supply chains get violently redirected. The invaders hijack the conveyor belts. They force your factories to completely abandon their normal duties and instead mass-produce the exact raw materials they need to spread.
Chapter 2. Hijacking the supply chain, rewriting the factory orders. And let me tell you, these microscopic invaders are incredibly demanding bosses. To multiply, they demand some very specific metabolic loot. First up, they need an immense supply of nucleotides, which are essentially the raw letters of the genetic alphabet, just to copy their viral genomes. Next, they need lipids, or fats, to build protective outer envelopes for their clones. And finally, they demand absolutely endless amounts of energy, or ATP, to fuel this exhausting replication process. Because viruses like dengue or rapidly dividing cancer cells, well, they show up completely empty-handed. So they aggressively push your normal energy pathways, like glycolysis, into absolute overdrive, just to get a quick burst of energy. Then they hijack the TCA cycle, which is your cell's main engine to extract raw materials. They ramp up nucleotide synthesis to print their own RNA and DNA, and they stimulate lipogenesis to build new protective membranes. It's a total systemic takeover of your infrastructure.
Chapter 3. CAD, the double agent, the ultimate inside man. Now we meet the star of the show, CAD. To put its size of 1.5 megadaltons into perspective, if normal proteins in your body are like individual workers on an assembly line, CAD is a sprawling, multi-story industrial complex. In the microscopic world, a megacomplex of this size is a massive piece of machinery. It commands huge amounts of resources and dictates major operations inside your cellular factory.
Normally, CAD is just a hard-working foreman. Its main gig is governing the synthesis of pyrimidines, those essential raw letters of the genetic alphabet we need for healthy DNA and RNA. It's a super vital, constructive role. But researchers recently figured out that this massive enzyme is actually leading a double life. It can be flipped into a highly dangerous asset for the enemy.
You see, aside from its normal construction job, CAD has a hidden capability. It acts as a protein deamidase. Now, think of a protein deamidase kind of like a master hacker rewriting a security protocol. By chemically flipping the function of other proteins, specifically by popping off an amide group, CAD can turn off your cell's internal alarm systems while simultaneously cranking up the production lines. It is literally the perfect tool for a cellular burglar.
And the way this heist is executed is just a perfect four-step tactical operation. Step 1. The virus breaches and enters the cell. Step 2, the virus deploys a specialized protein to actively switch on that CAD megacomplex. Step 3, CAD uses its hacker-like ability to deatomate and completely disarm the cell's immune alarm, which is known as RELA. And then Step 4,
With the alarm muted and security totally down, the cell is forced to build viral clones without any interruption.
Chapter 4. The immune system strikes back, activating internal security.
Thankfully, our cells do not go down without a fight. They actually employ a literal scorched earth tactic. Rather than feed the invader and let it spread, our innate immune system calls in specialized security forces called kinases. These kinases are deployed to deliberately sabotage our own hijack factories. Your cell essentially decides it's way better to break its own machinery and starve than to hand over the keys to a virus or a tumor.
This counterattack happens in a highly orchestrated, rapid sequence. In mere milliseconds, internal sensors detect the invading pathogen. Within minutes, elite security kinases, TBK1 and IKK-beta, are deployed into the cytoplasm. Over the next few hours, they hunt down the compromised CAD double agent, chemically modify it, and completely disable it. And by doing this, within a matter of days, the viral or tumor supply chain is completely starved of the vital nucleotides it desperately needs to survive.
Researchers have pointed out in recent studies that innate immune activation dramatically impairs CAD's enzymatic activity. Basically, that means pyrimidine synthesis is directly shut down by our own immune response. Simply put, our immune system doesn't just chase the pathogen around with antibodies. It literally rewrites the metabolic landscape. It cuts the power to the very factory the pathogen is trying to use, stopping the disease dead in its tracks.
Chapter 5. A new medical arsenal. Cutting the power to hijacked factories.
For decades, medicine traditionally hunted the virus directly, but viruses mutate rapidly, which can render older drugs completely useless. Our own metabolic factories, however, they don't mutate. And this has led to a massive paradigm shift. By creating therapies that target our own host enzymes, instead of hunting the invader, we can chemically cut the power to these hijacked supply chains. It's a much more robust, mutation-proof strategy to sensitize tumors to chemotherapy or to stop a replicating virus cold.
We actually now have a specific cavalry of drugs designed to deliberately starve these hijacked factories. For instance, PCAIs and compounds like 2-TCPA directly target the CAD double agent to disrupt nucleotide synthesis. That blocks both tumor migration and SARS-CoV-2 replication. Then there's orlistat, originally an anti-obesity drug, now being utilized to halt the lipid synthesis needed for viral envelopes. And we have STING agonists, which trigger those elite immune kinases we talked about to shut down CAD and amplify the power of chemotherapy.
So, mastering this art of cellular starvation, it could literally cure the incurable. If we can defeat these invaders by safely manipulating our own metabolic machinery, it really makes you wonder, what other hidden double agents are hiding out in our cells right now, just waiting to be discovered?